Hello, my name is Nora Fritcher, and I'm an undergraduate researcher in the Ovarian research group in the Department of Chemical Engineering and Material Science. My research for this year op was the cell cycle position dependence of dormancy induction in ovarian cancer cells. We were with ovarian cancer cells, and a substantial obstacle in ovarian cancer treatment is the high recurrence rate, which is hypothesized to be the result of dormant cancer cells. This subpopulation of cells can temporarily exit the cell cycle and exist in a growth-arrested state, the G0 phase, in response to harsh external conditions. This allows these cells to evade the toxic effects of common chemotherapeutics, which target rapidly dividing cells. After treatment ends, dormant cells can then re-enter the cell cycle and resume rapid proliferation, leading to disease recurrence. The Exxon group previously developed a silica-based gel which induces dormancy in cancer cells by physically immobilizing them in a stiff matrix that inhibits cell motility and proliferation. As seen in the bottom left, only dormancy-capable cells can survive within the gels. The image in the top right shows what the silica gels and encapsulated cells look like under a microscope. Ki-67 staining, shown in the bottom right, is used to assess cell proliferation. The blue is DAPI, which stains the nuclei of cells. The red stains Ki-67, which is a protein only expressed in the S, G2, and M phases. We use Ki-67 staining as an indicator for dormancy. Dormant cells are in the G0 phase and thus do not express the Ki-67 protein. Blue cells are dormant cells, and red/pink cells are not dormant. As you can see, in the day 0, the first day the gels are encapsulated, there are a lot more pink cells, but by day 3, those cells have died and most of the remaining cells are dormant. Serum starvation has been shown to synchronize cells in the G0 phase. As seen in the diagram, cells are normally cultured in 10% FBS supplemented media. FBS stands for fetal bovine serum. This media is removed and then serum-free media is added. Waiting for 3 days enriches cells in the G0/G1 phase. After reintroducing serum, cells re-enter and progress through the cell cycle together. The graphs along the bottom of the slide show the cell cycle distribution of 2 different kinds of cells that were synchronized using serum starvation. At the 0-hour time point, right after serum starvation, most of the cells are enriched in the G0/G1 phase. After reintroducing serum, the later time points show the cells progressing through the cell cycle together. Here are the results from some of my research. Varying the recovery time after serum reintroduction allowed for the enrichment of cells in different phases of the cell cycle. This data was collected by fixing the cells, staining them with rubidium iodide, and running them through flow cytometry to create the histogram seen in the middle of the slide. These synchronized cell populations were then encapsulated to evaluate their ability to enter dormancy. The cells that were serum starved for 3 days with no recovery were enriched in the G0 phase. These cells also showed improved survival when encapsulated in the silica gels, suggesting that cells in the G0 phase can more readily transition to dormancy. However, the cells that were serum starved for 3 days and allowed 1-day recovery were enriched in later cell cycle phases such as the S, G2, and M phases. These cells showed worse survival in the gels, which suggests that cells in later cell cycle stages cannot transition to dormancy as easily. In this set of experiments, cells were allowed a longer recovery period after serum starvation. As seen in the table and the 4th histogram, cells that were serum starved for 3 days and allowed for 3 days of recovery have a similar cell cycle distribution to the control cells. Additionally, serum starved cells with 7 days of recovery show a similar survival in the silica gels to the control group. These results suggest that eventually cells return to normal after 3 to 7 days of recovery because their cell cycle distribution and survival rate in the silica gels are very similar to the control. This indicates that changes in survival rate in the silica gels immediately after serum starvation and 1 day after serum starvation that were mentioned in the previous slide are in fact due to cell cycle position, not because cells are physically altered in any way by serum starvation. To conclude, varying the recovery period after serum starvation allowed for the enrichment of cells in different phases of the cell cycle. Cell cycle distribution of the serum starved cells eventually returned to normal indicating that changes in the survival rate during encapsulation were due to cell cycle position. Cells enriched in the G0 phase showed higher survival when encapsulated, while cells enriched in the S and G2/M phases showed worse survival. This suggests that cells in the G0 phase can more readily transition to dormancy than cells in later stages of the cell cycle. Future experiments will be done to investigate the survival of dormancy-capable cells when re-encapsulated during different cell cycle phases. As seen in the graphs below, it has been shown that dormancy-capable cells extracted from the gels display improved survival when re-encapsulated. We are interested in knowing if it is possible to push these extracted cells into sensitive cell cycle phases where they cannot transition to dormancy as easily. If yes, then this suggests potential vulnerabilities in this population that could be exploited for future cancer treatments. Thank you so much for taking the time to listen to my presentation, and I would like to thank Samira Azarin and Andrew Fredrickson for their help and support this past